Hello, my name is Yunseol Park, and today I'll be presenting about translation initiation site prediction using deep learning and synthetic datasets. Translation is a biological mechanism that dictates protein formation. Therefore, translation initiation site, or TIS, is an essential site to predict for genome annotation. There have been many advancements of machine learning technologies for such annotations or predictions. And the understanding of the important feature is important as this may aid in uncovering translation mechanisms, among other things. However, many of these models are black box in nature. To deal with this, we made use of a synthetic dataset as they are easier to manipulate compared to real data. This is the overview of the methodology. The datasets we used are the synthetic, the real, and combined datasets for the main part, and we used human dataset for transfer learning. The synthetic dataset was generated using 5 features: the consensus sequence, the upstream ATG, the downstream stop codon, donor splice site, and the nucleotide frequency. An additional feature was used named codon usage, which replaces nucleotide frequency in this workflow, and it adds codons instead of nucleotides. The models SBBM, RBBM, and CBBM were trained using the synthetic, the real, and the combined datasets respectively. Here we can see that CBBM has similar prediction effectiveness as RBBM. And as CBBM would have learned features from both the real and synthetic datasets, we can say that the features of SBBM may be overlapping with those of RBBM. This is because if SBBM and RBBM learn from completely different features, then the effectiveness of CBBM would also have to be very different from the two. For feature analysis, we made use of occlusion. The occlusion of the feature of interest and the occlusion of all features except for the feature of interest. On the left, in the left table, the lower the value of the F1 score, the higher the influence of the feature. For the table on the right, the higher the value, the more influential the feature. And in bold, I have indicated the reference points. And from the results, you can see that the consensus sequence and downstream stop codon are positively influencing features, along with nucleotide frequency. On the other hand, codon usage is an unexpected result. It has a very low F1 score. One possible reason for this is that the model might be able to learn different k-mers and not just 3-mers or codons. We also generated sequence logos for the consensus sequence. At positions -3, +3, and +4, we see that the consensus sequences for all 3 models are very similar, or exactly the same. The only difference are the contribution scores. For example, cytosine at position +4 is much higher at for RBBM than for SBBM or CBBM. We also performed noise analysis. Specifically, we performed class noise analysis and attribute noise analysis. For attribute noise, we consider the synthetic features as incorrect features. We made use of ELA, or equalized loss of accuracy, as the metric. Here, the lower the ELA value, the better the model behaves with the given noise. On the left graph, you can see that there is a high spike of ELA value for SBBM, regardless of the noise level. On the other hand, CBBM does not show that same spike. Instead, it has very similar ELA values as RBBM. In the graph on the right, the black line indicates the reference line, and it indicates the SBBM without any noise. The consensus sequence at noise level 0% is already higher than the reference point. And in this, we can say that the removal of consensus sequence is thought to be a noise for the model. And you can see that the ELA value decreases as we add the consensus sequence. Another thing that we can see is that there is a high spike of ELA value for codon usage at noise level 20%. Finally, we performed transfer learning. Using the 3 previous trained models, we froze some of those layers and trained human data. Without any pre-trained models, we observed overfitting. But the use of CBBM reduced this overfitting and increased prediction effectiveness. Of course, RBBM showed better results, but as CBBM was trained using the synthetic dataset as much as the real dataset, it has more potential, since we could create a better and more generalized synthetic data to train a pre-trained model that could be used for many other use cases, many other species. In conclusion, SBBM seems to learn from similar features as RBBM, albeit in different magnitudes. And CBBM has similar effectiveness as RBBM, and its use reduced overfitting when training with small data. Among the features we studied, consensus sequence and nucleotide frequency have the most significant positive influence. On the other hand, codon usage has a negative influence. To improve this idea and project, codons could be further investigated. Furthermore, a more sophisticated and generalized synthetic data could be generated to create a pre-trained model which could be used effectively for data-insufficient use cases. That is the end of my presentation, and thank you for listening.